Hi, I'm going to be going over the Genes and Environment lab, which is two parts. So for part A, you're going to be watching this video. The link is in the PDF right here. And you're watching a video called Ghost in Your Genes that's made by Nova. And the answers, I tried to help you out by putting the time. So this is at 45 seconds. It starts answering this question. At 2 minutes and 17 seconds, it starts answering this question. So in case you get stuck or lost, or maybe you want to just do, the first half, and then you want to come back tomorrow and do the next half, you'll kind of know where you left off. I don't want you to just jump from one to the next because there is a lot of information that is given after this number, but it just sort of shows you where to kind of look if you're having trouble finding the answer. All of the answers are in the video except for this one, which was kind of strange. And so if you're not sure what the answer to this question is, it's actually answered in the first couple seconds of this video. What is epigenetics? So you can jump up there, go ahead and watch that first, and then you can answer question one. If it feels like there's a couple questions you're not sure about, or it seems to be really confusing to you, I don't want you to stress out. I'm going to give you full credit if you can correctly answer 15 of the 20 questions. I would still like you to try to answer all 20, but if something is kind of off of your question, your answer, or you miss the mark on it, I'm not going to mark that off for 5 of them. But it's a fascinating topic about how different things can affect our DNA, the DNA that we're born with. For part B, and I'm going to open this up in Cami, for part B, you're going to actually be creating a model that shows how DNA can be turned on or off, genes can be turned on or off. So DNA is this long, coily double helix thing, but in reality, it's wrapped around histones, and we're gonna be using cotton balls to represent our histones, and we're gonna use some rubber bands to represent proteins that are holding the histones in place. So DNA needs to be kind of organized and coiled. When it becomes a chromosome, it coils even more. And part of the way it's packaged is with these histones. And histones are also involved in whether a DNA turns on or off, whether a gene is expressed or not expressed. So they can activate it or they can deactivate a gene. The gene's still there, but the histone can affect whether it's activated or not. So it says to go ahead and make this. If you have these supplies, that would be great. You can go ahead and make it, and I'm going to show you how I made mine. But basically you need cotton balls, small rubber bands, and string. It doesn't have to be plastic string. Okay, so here's my attempt. So I've got 1, 2, 3, 4. I'm supposed to have 6. I actually ran out of thread, so I had to tie a knot and add more thread. So I'm supposed to wrap the string around twice and then put a little rubber band to hold the histone in place. So these are just little hair rubber bands I found at the bottom of a drawer. And again, if you don't have perfect materials, you don't have to sweat it. Okay, so it just makes it a little more visual if you can do it yourself, but you could watch me or you can look at the pictures on the lab. Okay, so here's our 6th histone. All right, beautiful. Okay, so there's our 6 histones. Let's see what we're supposed to do. All right, so this gene is supposed to be um, preventing cancer from forming. So if it's on, then it prevents cancer from forming. But if part of the gene gets turned off, then it allows cancer cells to grow. So we don't want parts of this gene to get turned off. Okay, so we've completed the gene. We're supposed to draw a picture of it. You can obviously just draw a picture of this. Okay, now here are the following environmental factors we're going to be flipping a coin for. And they're going to affect whether the gene is turned on or off. So if we flip coins and they're both heads, then no change is going to happen, and we're going to leave our histone and our protein as is. And if we get one head, one tail, then we're going to cut off our little protein. And if it's two tails, then we're going to take out our cotton ball or histone. So we're going to go ahead and flip for nutrition. That is going to affect the DNA switching on or off for the baby in some ways. Her stress is also going to affect the gene expression, whether she's exposed to smoke, the nutrition of the child, the stress of the child, and smoke exposure. So a lot of these are affecting our DNA when we're developing as a baby, unborn baby, and also as a young child. So let me go ahead and get my coins. Okay, so we're going to go ahead and flip first for the mother's nutrition during pregnancy. So let's go ahead and flip for that. Okay, two heads, which means I'm not going to make any changes. She ate healthy foods, yay. So that's going to be okay for mom, okay for baby. All right, so I was just going to do one example, but since she came out both heads, let's try again for the second. Whoa. Okay, two heads again. I'm going to just, oh. All right, I'm going to, oh my gosh. Okay, look. So it came up heads and tails, which means this is the mother's stress during pregnancy. She had some complications, which caused some stress. And that is affecting the baby's genes. So here we're going to cut off the protein. Okay. All right. And shall we do one more? Okay. Let's pretend I flip for smoking. Okay. And poor baby, mom smoked during your pregnancy. And so for two tails, we're going to have to remove our histone. Okay. So you're going to go ahead and continue down the line. We did 3, and you're going to do the other 3. And you're going to write your results. So you can just go ahead and write in heads, heads for here, no change. Here we had heads, tail, and then this one we had tail, tail. And then we're going to draw our finished one. And if you don't have all the materials, you can sort of just, you can just sort of redraw this one, but with the changes that you see, the changes that you made. So you might have to make little notes and say, no rubber band, no no cotton ball, no histone. Okay, and so remember, the more changes that you have, if there's 0 to 2 changes or errors, the individual has a very low chance of getting cancer because the gene is still going to be on. But if you have 3 to 4 changes, you have a higher chance. 5 changes, it's going to affect the gene expression more. The gene is still there, but the fact that the histone is missing or the protein is missing is going to affect whether it turns on or off. Okay, and then you're going to answer these questions right here below. All right, if you have any other questions, feel free to contact me. Otherwise, I hope you enjoy this activity.